Alright guys, to continue on where we left off last week, I would just like to explain a bit more on homologous recombination. So if you remember on the topic of transgenic mice, usually it's recommended, especially when you want to produce knockout mice, that you get your gene to be in a homologous state. So more or less, okay, this is why. So but first, okay, let's have a look at how you can integrate or insert your gene using any method. So most of the time it can occur either by homologous, which is denoted by H, or non-homologous, NH recombination. So this recombination occurs a lot during mating, and this is a quite common occurrences in nature. Actually you couldn't control it, okay, but when you are manipulating or when you are proceeding with recombinant DNA, you could do something about it. Now, I'm not going to explain more on the differences between them. Probably I'll find a link and basically put a video on in e-learning for you to look at in case you have forgotten. You have learned about homologous and homologous recombination in cell molecular biology in your first year. So frequency of non-homologous is actually precedes homologous by about 5,000-fold, and this is quite common even in Asia. So if you want to get homologous recombination on integrant, okay, which of course, like I said, needed for knockouts, you have to have a selection scheme for those, which means that you need to have a very targeted method and approach. So let's have a look at an example of a transgene for producing transgenic animal with incorporated into a vector. So your transgene is here, and it is basically in between, okay, two homologous region here, and there are genes, okay, that codes for TK1 and TK2. They are herpes simplex virus thymidine kinase genes that make cells susceptible to ganciclovir. In nature, ganciclovir causes eye infection in animal. And you have here, okay, which is a marker gene, neomycin, which is by right, okay, if it's integrated, then your cell would be resistant to neomycin. Now, let's take a look what happens when you have non-homologous recombination, which occurs a lot. So you have your vector over here, and you made it, okay, with a non-transgenic. So what you get, okay, is actually bits and pieces for everything. So as you can see, denoted by the white are the ones, okay, from the vector early on, and the ones, okay, in black is actually from the second or the mate. So you can see that it gets every bit and pieces for everything. That's what we refer to as non-homologous recombination. Every bits and pieces gets it. So this is what happens, okay, when you perform screening. Your transformed cells, okay, are neo-resistant because neomycin is there, but it will be ganciclovir sensitive. Why? Because both TK1 and TK2 are there. So to make it ganciclovir sensitive, actually only one TK is needed. And you will see it here again in homologous recombination. Same vector, okay, and same mate. And you can see that not all of them are transferred into the new recombinant. So if DNA goes by homologous recombination, transformed cells will be both neo-sensitive and ganciclovir resistant. Neo-resistant and ganciclovir resistant. So in this case, you can use a double selection to make sure that your homologous integrant is actually quite good, and you can actually be firm and very good verification out of it. So let's take a look at them side by side. So random is non-homologous, targeted is homologous recombination, and you have your vector over here. So remember your TK, your neo, and basically your transgene over here. Now, if you were to have a random gene insertion or non-homologous recombination, this is what you get. Like I said, every bits and pieces gets integrated one to another. But if you have, or if you go for the targeted gene insertion, this is what you will get, the result, okay? Not all of them will be integrated together, but again, you get some of them to be integrated. So for this one, okay, the cells are resistant to neomycin and will be killed by the ganciclovir. But okay, over here, for your targeted gene insertion, your cell will be resistant to both neomycin and ganciclovir, which is actually what is needed for knockout. Now, to explain knockout and how you can more or less design an approach, this is how you can do it. So you have your target gene, and basically your target gene has been inserted into your vector of interest. So most of the time, transgenic animal vector, it has TK gene. That is actually one of the criteria to determine for the screening purposes. And you need to insert neo gene into it. So when you do your insertion, can you see that the neo gene is inserted in between the target gene? And hence, remember back in transgenic plant, there are lots of ways of you to do this. So you transform the KO or knockout plasmid into embryonic stem cells, and you can perform double selection because by right, okay, if everything goes well, your knockout gene should be resistant to both neomycin and ganciclovir. And if you have confirmed okay, that the knockout gene okay, is both resistant to neo and ganciclovir, you can actually just inject it okay, into a blastocyst and proceed with producing your transgenic animal. Alright, so there you have it. I would say then proceed with the second second more or less topic okay, which is on knockout mice. So knockout mice is basically a common way to more or less determine how gene would function when a certain gene is turned off. So more or less okay, it is defined as containing the same mutation artificially introduced because you purposely switch off the function of a certain gene. So okay, it basically abolishes the activity of the original pre-selected gene. So knockout mice would result in mutant phenotype that may provide clues of normal gene role. Can the gene perform as normal if it's being turned off? And certain human diseases caused by the loss of function of certain gene are usually studied using these knockout mouse models. And the key principles are you need to have a mutant version of the pre-selected target gene constructed in the lab. The knockout has to be achieved by swapping the functional copy of the gene for mutated version in mouse embryonic stem cells. Mice are derived from modified embryonic stem cells, and the difference okay, between the knockout technology that we have now with traditional constitutive knockout mice is that mutation is present throughout development and in all cells of the adult. In traditional, no, it's only at a certain stage and developmental processes. So as you can see. Alright, the production okay, of knockout mice is of course through gene targeting as you have seen in earlier part to be done by homologous recombination. This is done by replacing the resident sequence with a modified related sequence, your transgene of interest. Replacement by homologous recombination. And the whole gene targeting processes would have to take place in the embryonic stem cells again. And embryonic stem cells are obtained from very early male mouse embryo normally, whereby later it could be differentiated because they are pluripotent, remember, into all cell types upon integration with another embryo. Now, why do we do? Okay, I've actually said it in 2 previous slides earlier on, okay, but the aim of knockout mice usually is to get modified embryonic stem cells to contribute to the germline for sperm, because some sperms produced might carry the desired mutation, and fertilization will of course give rise to a copy of the gene in every cell of the offspring, which means that it could be integrated and inherited in the offspring and the next generation. So you need to do some interbreeding, and of course interbreeding will produce homozygous individuals carrying 2 copies of the mutant gene, and these are referred to as knockout mice. So remember your pedigree from last week, so all those are being incorporated in. So these are the steps to produce knockout mice. More or less they are similar to producing transgenic mice, but with some modification. So first and foremost, you need to isolate developing embryo at blastocyst stage. So usually the color of the fur of the mice is important, and for this step they use a gray fur mice. So this is as you can see if you remember when I was telling you earlier on. So this is your embryo, this is the blastocyst stage, and this is the inner cell mass. Second stage, you remove embryonic stem cell from the gray fur blastocyst, grow the stem cells in tissue culture, and basically this is how it is done. You take the embryonic stem cells from the blastocyst, the inner cell mass, grow it in the lab. Third step is to transfect. In animal cells, we call it as transfection instead of transformation. So you transfect stem cells with homologous recombination construct, and then later on, you select for homologous recombination by growing stem cells in both neomycin and ganciclovir. Remember in earlier part of this video that you need to make sure that it is both resistant to neomycin and ganciclovir. So mutant version of the target gene is introduced into the embryonic stem cells, and of course, by homologous recombination, it would swap some of their mutated version, as seen by the representation of the diagram here. Now, the fourth step is to remove the homologous recombinant stem cells from the petri dish and inject it into another blastocyst that would have come from white fur mice. So you use a different color. This is very functional later on, as you will see. So embryonic stem cells again need a host embryo to develop, and this is usually done with a mouse of a different colored fur. Alright, so the fifth step is to implant several of the chimeric blastocysts into pseudo-pregnant white fur mouse. So the surrogate mother is of a different color than the original blastocyst, so white. So the use of mice with different coat color for the embryonic stem cells and the host embryo allows the mutant gene to be followed phenotypically. So as you can see over here. The sixth step okay, is that, of course, and hopefully, if everything goes well, the mother will give birth to mice. So you will see that it will give basically birth to a wide range of mice color. So some of it would be normal white fur, some of it would be chimeric mice, some of it would have fur, some of it would have kind of like spots or combinant of the gray and white. So those are the chimeric mice. So the chimeric mice usually, by the phenotype, okay, we thought that probably they may have the integrated gene, but it needs to be more or less tested and verified further. So this is basically what happened. So remember, surrogate mother is white in color. So it produces again lots of offspring with different fur color. So some of it would be white, okay, some of it again would have spots, a combination of both white and also the original gray. Now the seventh step okay, is basically to establish the transgenic line. You mate the chimeric mice with the wild type white fur mice. So remember, this is the basic from your breeding pedigree. So if the gonads of the chimeric mice were derived from recombinant stem cells, all the offspring will have gray fur, and every cell in gray mice are heterozygous for the homologous recombination. So this is actually one of the limitation and something the researcher needs to remember later on. Okay, so this is what again it will produce. So now in second generation, all of them will be more or less quite similar in color. Okay, so kind of like brownish or grayish and whatnot. So how do you know which one actually carries the gene? So you need to cut off the tip okay, and test, do some assays. So the eighth step okay, is also in the breeding pedigree to make the heterozygous gray mice second generation and genotype the gray offspring. You mate them with one another. Now okay, it's basically to mate okay, with the transgenic animals. So you identify recombinant homologous breed and breed them to produce a strain of mice with both alleles knocked out. So the pure breeding mice strain is known as a knockout. So interbreeding, of course, produces knockout mice okay, by right because we want both of the copies okay, to have the knockout gene. So this is actually how it is done. Again, following similar Mendelian genetics, okay, the postulation of 1 to 1. Alright, so based on what has been done on knockout mice, this has been done over years, so two observations were basically seen. And the mice okay, are unaffected by their deficiency, so redundant for a single missing pair of alleles as many genes appear not to be indispensable. So if they are heterologous, usually they are not really affected okay, by one of the alleles being turned off. So most genes are pleiotropic, which means that they are expressed in different tissue in different ways and times during development. This is actually why. And the phenotypes of knockout mice or mouse okay, usually provide important clues about normal role of a certain gene, especially if you want to see of how it would behave, whether it will be really affected if it's being turned off. So this would of course be useful as it could be used to investigate the biochemical and physiological aspect of human diseases. So I think I'll stop here and proceed with the next video because then it will be too long and it will take a long time to upload.